With that, we will turn to our third speaker of the afternoon, Thomas Perls. Dr. Perls is a professor of medicine and geriatrics at Boston University School of Medicine, a geriatrician at Boston Medical Center, and the founder and director of the New England Centenarians Study, the largest study of centenarians and their families in the world. He attended Pitzer College in Claremont, California, and then the University of Rochester School of Medicine, followed by the Harvard University School of Public Health for his master's in public health, majoring in quantitative methods. His research aims to decipher the genetic and environmental factors that contribute to longevity. He's among the international leaders in the field of human exceptional longevity and has been responsible for numerous novel and pivotal findings in the field. He's also a principal investigator in the NIH Institute on Aging funded Long Life Family Study. He's the author of the online Living to 100 Life Expectancy Calculator, something that may be of interest to any number of those who are with us this afternoon, as well as several academic articles about the medical misuse and illegal distribution of growth hormone and anabolic steroids, including testosterone, for anti-aging and bodybuilding. He's won numerous awards for his work on aging and gerontology. And with that, I will turn the podium, the kind of virtual podium, over to Dr. Perls for his talk, Centenarians, a human model of resilience, even resistance. Dr. Perls.
Thank you very much, Bob. And my thanks to the Society for having me is a real honor. Thank you. So I think Jacob is going to pull up some slides for me. Great. Thank you, Jacob. So the title is exactly as Bob read it to you all. Next slide, please. So the New England Centenarian Study was started quite a while ago now, 1995 by me, really in the midst of my geriatrics fellowship at Harvard, when I had the chance to take care of a couple of centenarians. And, you know, during my training and as a medical student and then as a resident and then fellow, this idea of the older you get, the sicker you get, is really quite prevalent in medicine. And it was something that we would experience in the hospital, in that our oldest patients ended up being the ones we were most worried about in terms of the consequences of whatever they were coming in with. But then I had these two centenarians that were on my patient panel. Really, if it was a matter of the older you get, the sicker you get, I expected both of them to certainly have dementia. A lot of people felt that given that half of people over the age of 85 had dementia, now let's not forget that half of people over the age of 85 do not have dementia. But having said that, and that the prevalence seemed to greatly increase with older and older age, that by the time you were 100, we'd expect everybody to have dementia. Well, these two people that I had on my panel definitely did not. One was playing piano concerts on a weekly basis, very difficult Chopin and beautifully, all from memory. The other was and she was 102. The other was a guy who was now volunteering in a occupational therapy group, mending people's clothes. He had been a tailor all his life, and was teaching young and old about tailoring and so on. When he wasn't doing that, when he wasn't active doing that, he was robbing the cradle, dating his 85-year-old girlfriend. To say the least, these two people jarred me a bit, made me very curious as to whether other centenarians were like that. And now 20 years later, I'm still up to no good studying these folks. These are just a few pictures. One is one of our research assistants, Stephen, with the same guy over on the far right. This man was showing a certificate here with his daughter and son-in-law. By the way, the daughter is in her early 80s. He's 106, and that's a certificate from Patton's army that he was a part of at the Battle of the Bulge. And he had amazing firsthand accounts, I must say. One of the wonderful things about studying these folks and meeting them is that they are literally living historical treasures, and you get firsthand accounts that just blow you away. It's a wonderful, it's been a wonderful experience meeting these folks, and just that aspect has been great. Now, I have here some of the studies that are funding our work from the National Institute on Aging, the Longevity Omics Study, the Long Life Family and the Longevity Consortium, Centenarian Project, and more recently, Jacob hit the space key. One more study coming up on the slide, Jacob. I think if you, there you go. We just got this $20 million study that we proposed that we titled RADCO, or Resilience and Resistance Against Alzheimer's Disease in Centenarians and Offspring. And that study really is in part the impetus for the title of this presentation, and I'll get to that particular topic towards the end. Next slide, please. So what's special about centenarians? People who live to 100 are certainly rare, but those who live to 110 and older are super rare, and in fact are called supercentenarians. 100-year-olds are about 1 per 5,000 in the population in well-to-do countries, and 105-year-olds, what are termed semi-supercentenarians, are about 1 per 250,000. And then we have this very, very small group of people, 110 and older, that are termed supercentenarians, or 1 per 5 million. At any one time in the United States, we have about 60 to 70 supercentenarians. We have about 50 to 70,000 centenarians alive in the United States. Next slide, Jacob. This is just a picture. Oh, there, sorry. This is a picture of Sarah Canals, our oldest subject, who was also the oldest, second oldest in the world at the age of 119. And she's here with her great-great-great-grandson. We visited her when they had gathered these 6 living generations all at once. And what was fun was Sarah was there with her daughter, and her daughter lived to be 100. And so one of the few instances where we had both the mother and the daughter in our centenarian study. But that's a truly remarkable family. Next slide. So what about what's it like living to 100, and what does it take to get there? People age very, very differently from one another. All you have to do is look at your neighbor who may be the same age, and how are they doing, both physically and cognitively, or their parents for that matter. But as we get beyond the age of 100, it's very, very interesting that people become more and more alike. Now, as far as, is it a good thing to live to 100? Some people, ascribing to this idea, the older you get, the sicker you get, may say, "Oh, I'd never want to live to 100." But in fact, what we have found is that about 90% of centenarians are disability-free up through their early to mid-90s. And so they even may have age-related diseases, some of them starting at 60 or 80, but they deal with those diseases much, much better than others who might otherwise die from those diseases. And they are living, again, independently up through their mid-90s, which I would say most of us would embrace and love. And instead of it being the older you get, the sicker you get, our work is showing that it's the older you get, the healthier you've been, which I would wager is a very optimistic, positive view of aging. Now, 15% of our centenarians are what we would call escapers, who at 100 really have no evidence of aging-related disease, no dementia, perhaps maybe a little bit of osteoarthritis, some wear and tear, but otherwise truly amazing. And it's these escapers that are the ones that go on to be, many of them go on to be to much older age. In fact, our cognitively intact centenarians are virtually immortal. And it's only when they start to show some cognitive impairment that we begin to worry about them. It's our number one marker for mortality in these individuals. But if you look at this group, and then you look at centenarians, supercentenarians, 70% of them were what we call these escapers. And on average, they're both disability free at 106. But in addition, they don't have these age-related diseases become manifest until much later, around 105 or 106. And this is where they are just so alike in terms of the presentation, incidence, and prevalence of disability and diseases. And if they are so alike at these extreme ages, then the thought is they must have some things in common that are enabling them to get to these very old ages. Next slide. So if you look at people, say, to about age 90, which is already an amazing age. It's 30 years beyond the age of 60. I would wager that most Americans, most people have the genetic makeup, have the average blueprint, if you will, to get to 90. And the reason we know this is if you look at the Seventh-day Adventists, for example, they have the highest average life expectancy of any group that we know of in the United States. It's a very mixed group in terms of race, ethnicity, geographic location, but what they have in common is this religion, and a religion that really considers one's body as a true gift from God, and that to do bad things to that body, to have bad health behaviors, really squanders that gift. It's a sin. And they have health behaviors that generally are very, very good for them. They don't drink, so maybe a little bit of alcohol is good for you. They don't smoke. They tend to be vegetarian. They regularly exercise. When they do eat, they tend to eat in moderation, which means that they usually tend to not be overweight. They tend to be at a healthy weight. And they also spend quite a bit of time with family and religion, which may help them manage their stress better than others. And so it's really the health behaviors that are explaining by, the real majority of variation of how old people are going to live to up to about that age. And if you do everything wrong, you're a couch potato, you're smoking, you're passive in your cognitive activities, meaning you're watching a lot of TV and not doing much else. You eat way too much red meat. I can go on, but these individuals, it becomes no surprise that they're passing away in their 60s and 70s. So again, a lot of the variation of getting to around 90 is going to be 75% of our health behaviors. But again, where you have these individuals who are so similar in their compression of disease and disability towards the very end of extremely long lives, we see it switched around. And it appears that about 75% of the variation living to these most extreme ages is genetic. And very interestingly, it isn't really the lack of, we all have the same genes, we just have different variations of some of those genes that distinguish us genetically apart from one another. But it doesn't seem to be lacking genetic variants associated with certain diseases, but much more that these individuals have some genetic variants that are protective. They slow aging and they decrease your risk for aging-related diseases like heart disease, stroke, diabetes, cancer, and Alzheimer's disease. The other important thing is most genetic studies of complex traits really require tens of thousands of individuals, their DNA samples, to try and find genetic variants that they have in common. But because so much of the expression of this trait, that is living to 105 and older, for example, seems to be strongly genetic, we think that having a sample size really more in the low thousands is enough to discover these genetic variants. I also say that getting to these extreme ages, as I mentioned before, is extremely rare. And the reason is it's a bit like winning the lottery. Getting one or two numbers is fairly easy. Getting all 7 numbers, getting that right combination of 7 numbers is of course extremely rare, and oh, how we wish it wasn't so rare. Same thing here. Getting the right variants and the right combination of these 200 or so genes is very, very rare. Each genetic variant has a very modest effect on its impact of longevity, but as a group, it can have a very, very strong effect. So go on to the next slide. Let's talk a bit about resilience and resistance. I told you that the genes seem to be protective. They're slowing down aging, and they're decreasing risk for aging-related diseases. And what we have also found is that there are centenarians who have markers of Alzheimer's disease, whether it be these blood markers that we're finding or that are becoming pretty good at predicting Alzheimer's disease down the road or being coincident with Alzheimer's disease, as well as PET scans, these very high fidelity functional MRIs as well are pretty good markers. And if we have somebody who has these markers for AD, but they're entirely cognitively intact, we would say that they're resilient, that they're doing something that despite the presence of AD, they're not expressing it, that they're still cognitively intact. Then we have individuals who are resistant to Alzheimer's. These are folks that show no evidence of AD by these markers, and when they pass away, there's no evidence of Alzheimer's disease on their neuropathology. And so they've got something going on there that is just absolutely preventing the disease. These individuals are quite a bit more rare, obviously. We have folks who may be resistant at age 80, and then they slip into a resilience phase where the markers start to have an effect on the tissue of the brain, but they're still somehow not expressing the disease. Let's go on. So this is a bit of a depiction about reserve. Down in the left corner has to do with brain reserve. This has to do with actual structural features of the brain. Brain size, the number of brain cells, or what we call a neuronal count, the number of the amount of synaptic density, which has to do with the connections between the brain cells or neurons. And these are all increased in people who have more reserve. It's like there has to be more of these things have to be destroyed before there starts to be a problem. On the other hand, people with traumatic brain injury, infection of the brain, some psychiatric illnesses, some developmental disorders, they have less brain reserve. And so it's going to take less of an insult for the person to demonstrate cognitive impairment. And then there's cognitive reserve, which instead of being more the structural, instead of brain reserve, which is more the structural component, cognitive reserve has to do with more of the functional component. And there are things that we know that increase cognitive reserve and would therefore increase resilience and decrease, would allow you to perhaps have some kind of neuropathology going on, but you get away with it for a longer period of time. Cognitive engagement, come to these talks. Cognitive remediation, which has to do with rehab. Education, not just years of education, perhaps a bit of just how difficult some of the things that we learn later in life are, like the real heavy lifters would be learning a new musical instrument or a new language, but also not sticking with just one thing, trying new things, which may involve different parts of the brain, becomes important. Leisure activities, nutrition, and social stimulation. These are all very important for improving cognitive reserve. On the other hand, there's things that decrease cognitive reserve. Sedentary or passive mental activities, which I mentioned, like being a couch potato and staring at the TV. Malnutrition, lack of education, loneliness or depression, and very importantly, less sleep can greatly impact upon, if you do have a neuropathological process brewing, when that transitions into actual clinical expression of cognitive impairment. Next slide. So with our centenarians, both our study, a study in the Netherlands called the 100+ study, have been looking carefully at the roles of reserve, resilience in the centenarians and their kids. This is just a bit of a depiction where the top line shows that, for example, with many of our centenarians, like I said, they're living independently up through their mid-90s. They have a high test scores for a prolonged period of time. And they have so much reserve that despite the onset of some kind of neuropathology, they really don't express any kind of cognitive impairment until quite late. And then when they do, there's a very rapid decline, as if there's kind of a straw that breaks the camel's back phenomenon. Again, I would say this is what most of us would want, whatever age we would live to, where we compress the time that we experience troubles towards the very end. Then there's other people who may develop Alzheimer's disease and not have this either cognitive reserve and brain reserve, where they might also have a bit of a lower cognitive test scores because of less education and other factors. And then they start to have a slow decline. And then at some point, they develop Alzheimer's disease. You'll notice with the upper line, Alzheimer's disease occurs in these people as well, but not till much, much later. So the upper line is really the story of centenarians for the most part, and in their kids as well. The next slide. And here is some studies that we've done looking at the kids. And I'm not talking about children in terms of their age group, but the children of our centenarians who are generally in their 70s and 80s, few in their 90s. I told you about one who was 100 while their mother was still alive. And we see the same thing happening where they maintain good cognitive function over a longer period of time. And then when they do have impairment, it's a more rapid decline towards the end of their lives. Next slide, please. So with these various studies, sorry, the RADCO didn't show up there, but we are doing a whole bunch of data collection and data generation. We do very careful cognitive function testing. Now because of COVID, we've been doing it via Zoom meetings with our subjects. We get a week's worth of physical and sleep data with special watches that folks wear. We get very careful pedigree and broad pedigree data on everybody, looking at what ages they died at and what they died from, detailed dental and medical history, sleep habits, as I said, dietary habits intake. We do brain MRIs on our folks. Biological samples include blood samples. We're collecting fecal samples. You've probably heard that the microbiome has become of extreme interest in terms of the bacterial populations in our fecal sample, in our guts, as well as elsewhere on our skin, our oral flora, and so on. But that the substances produced by these bacteria and the interrelationship between these bacteria and the cells in our body really can be a symbiotic relationship that can have very, very important mechanisms for both healthy aging and unhealthy aging. We are collecting blood samples for what are called induced pluripotent stem cells, which is a very exciting area of research for us, where we're producing these stem cells from just our regular blood draws that can then be differentiated in the Petri dish into brain cells, for example. Once we have those, we can start to do studies to try and understand how the brain cells of, say, supercentenarians are different from the brain cells of people with Alzheimer's disease, and what are the biological mechanisms that are different for very specific kinds of brain cells called microglia and astrocytes that could very well be playing a really, really important role in either the causing Alzheimer's or preventing Alzheimer's. And then we also get brain donations on our subjects when they pass away, so that we really get a good idea of what, if any, pathology is going on. We can see who is actually resistant to Alzheimer's, who is not. We can send pieces of brain to our colleagues who do DNA expression studies from brain tissue. There really is almost a limitless amount of studies that we are doing and that we are sharing our data with really investigators around the world. Next slide, which is our last slide, I believe. So I've mentioned where we're going pretty much already. All of this is not to get a lot of people to 100 or older. It's to understand how these very, very special individuals are aging so slowly and are markedly delaying or escaping various diseases, with the hope that the discovery of those mechanisms that enable that will help us discover drug targets and discover drugs and strategies that would help the rest of us who may not have chosen our parents so well in terms of the genetics to delay and escape Alzheimer's, especially. That is the end of my talk, and would love to entertain any questions. Thank you.
Thank you very much, Dr. Pearl. So we do have some questions here. One comes from Dick Garwin, who is one of our older members. What is the evidence for 200 protective genes? Why not 50? What determines that number?
Well, we and others now have done these studies called genome-wide sequencing studies, where you look really at millions of genes and you look for genetic variants that these individuals have in common. And while there are many genetic variants that they may have in common, as I said, any one variant doesn't seem to do the trick in terms of a strong effect. And that would be expected, because both aging and a very complex, what we call phenotype or trait like longevity, is going to involve many, many biological defensive mechanisms, things that are combating free radical damage, for example, but also that excel in repairing DNA. Those are just two examples. Each one of those mechanisms is going to have numerous genetic variants playing a role in those mechanisms. And so because it's so complex, because there's so many mechanisms, there are going to be many genes. When we did those genome-wide studies, what we found was we created combinations of, we didn't create, we let the data drive the findings. And what emerged from those studies was that there were genetic signatures of close to about 200 genes, or these variants of these genes, that when combined did a really good job of predicting in a group of people who was 106 or older, for example, versus who was young. These signatures don't work so well when you're looking at people of 100, because the influence of genetics to get to 100 isn't as great as it is to get to these much rarer ages of 105, 106. So that's kind of the number that we are thinking. The other thing to know, one more thing, is that it isn't just one signature. People of different races, of different geographies, have different signatures to get to these ages because of the thousands and thousands of years that they evolved under different environmental exposure conditions. And so it takes different genetic variants for certain people to get to these extreme ages. So it becomes a pretty complex thing to understand, and it really speaks to the importance of why we have to study people of different genetic backgrounds to more fully understand what variants they have in common, what variants they don't have in common, what exposures are we talking about, so on and so forth.
Thank you. Howard Gardner asks, when one hears scientific findings, one can almost always explain them retrospectively. As a member of the research community focused on extreme aging, what's the biggest surprise to your colleagues, and which ones have caused the most controversy within your research community?
I think the first thing that emerged is back in the 90s and earlier, people thought that aging was so varied, so heterogeneous, you would never find genetic variants that seemed to distinguish one group from another. Now, with the explosion of genetic technologies, and perhaps even more importantly, the ability to analyze those data, and then actually finding subgroups of individuals where there does seem to be a pretty important genetic component. Now we are finding genetic variants that these individuals do have in common. And I think we are going to be able to start solving some puzzles about why some people are able to do this and others are not. I would say that was the biggest hurdle to overcome in what has now become an acceptable query line of science.
Thanks. Venki Ramakrishnan asks, has the number of centenarians been increasing with time? Were there centenarians in older times, for example, the medieval era or the Victorian era?
There were, much rarer back then. You know, when average life expectancy was 40 years old, a lot of people who could have lived to 100 didn't get there because they were dying of all kinds of infectious diseases and what are readily reversible problems. Every now and then somebody would sneak through, but very rare. Around around 1915, 1920, we really started to see huge changes in public health, clean water supply, the beginning of immunizations, other public health measures, better socioeconomic conditions, where at the time, up until then, we lost about a quarter of our children to infant-related diseases. Also, there were a lot of still pretty high mortality with childbirth. Once those problems started to really be dealt with, we were no longer losing a quarter of our population during infancy, and those individuals got to live on into adulthood. And some of those individuals got on to very old age. And it was really during the 1900s that we started to see, instead of centenarians being 1 per 100,000, they became fairly quickly 1 per 10,000 in the population. You add on to that much better care of older people in their 80s and 90s, and a huge change in the 1960s and 70s in cardiovascular disease rates, treatment of high blood pressure primarily, and then later marked decreases in smoking. We start seeing a few more centenarians. People.
Well, we have time for just one last question. And this comes from Linda Greenhouse. And she says, as one who reads the necrology at the start of every APS meeting, I'm always struck by the advanced ages at which our members die. Is the APS worth studying, or do we manifest your theories and findings so well that the results of such a study would be too obvious? Whoops, I think that we have that Tom Perls has frozen, and that we may not be able to hear from him unless he logs in again. I will wait about 30 seconds here to see if that happens. I think he's recognized that he has lost us here. I hate to go on like this by myself. I will mention one near centenarian and one almost actual centenarian among the APS members with whom I'm familiar. The real centenarian was the late Walter Monk, who many of you, I think, will remember, known as the Einstein of the seas, who died only a year or two ago. The other one who I knew personally for a while was Sewell Wright, the great geneticist. He died, he was my ideal because he died at the age of 99, almost 100, with a paper and press. And I will always hold that as kind of an ideal life. In any event, I think we're not hearing from Dr. Perls again right now, and so I think we will have to bring this meeting to a close without an answer to the great question that Linda just asked, and perhaps we can find a way to answer it again for all of us in some other way in the near future. In any event, I thank you all for joining us this afternoon and throughout this wonderful meeting. And I look forward to seeing you folks and perhaps others in person on the 28th to 30th of April 2022, when, if our luck holds, we will again be able to meet in person. So thank you all and good afternoon.